purposely put these panels together so that you can understand the life cycle development, as I was talking about earlier today, of medications, diagnostic devices, diagnostics and devices, my apologies. But before we would begin the two panels back to back, we have a new board member with us and also an award recipient that is going to be gone tomorrow night, so we wanted to make certain that we gave him the attention that he's due and also introduce him to you folks. Last year, we added new awards to our annual awards ceremony that were rooted in patient advocacy and education. These awards recognize key influencers who are working to inform all Americans about kidney disease, individuals and institutions advancing the health interests of kidney patients, and patient leaders who mobilize fellow kidney patients to make an impact on future health outcomes. We would now like to present the National Social Media Education and Advocacy Award to an individual who has established a significant reputation and following in social media and in the kidney community. Our award last year, our first award in this category ever, was given to James Myers, and many of you folks have seen James. He was transplanted this spring. He looks like $1 million. He mastered the medium of Facebook and YouTube in order to gather thousands of signatures on petitions demanding better access and better treatments for kidney care. He really set a model and a standard that we think a lot of folks will follow. Our recipient this year has mastered the platform of Twitter and is a true patient educator and very strategic in what he chooses to tweet and not tweet. He is currently president of Voice of the Patient, Incorporated, and also has spent much of his professional life within the renal industry at companies such as AbbVie, Astellas, and Pfizer. Kevin, you want to stand up for a second? Come on up. Kevin is a newly elected board member for AAKP, and his understanding of Twitter has led him to educate and train thousands of people on the power of this social media tool and the various social media platforms designed to maximize your voice as patients. We're pleased to present the 2016 Social Media Education and Advocacy Award to Kevin Fowler. Come on up.
Thank you very much.
Sure. Okay.
Thanks. I was surprised to get this, so thanks, Paul. And looking forward to being part of the board of directors here. Just one of my real passions is I had a preemptive transplant that Dr. Doug Johnson was talking about, and hopefully that's something that I can help him as well as the AKP increase that opportunity for patients to avoid dialysis like I did and have a kidney transplant. I'm 12 years and 1 month out, and I just like to see more people have the same experience like myself. So I'm really happy to be here. And I'm very excited to have the award, too. So go Twitter. Thanks.
So as you listen to these next two panels, I want you to keep in mind that nothing advances in kidney world without a group effort. And some of the folks that you're going to hear now are entrepreneurs. They're the drivers in industry. They're the drivers in American commerce who make it happen for kidney patients in terms of innovation. Nothing happens without those who have the courage and the resources and the ability to gather resources and investors to put new drugs, new devices, and new diagnostics out on the market. And the folks that you're going to hear from over the next couple hours are real leaders in their field. And when they get to a point where they need support, they'll often ask patients to support them. And AAKP very judiciously looks at that. But when we see something that's a good for the community and a potential breakthrough, we let people know. And I'm sure they'll let you know what their needs are on social media. Thanks a lot.
Hello, everybody. I'm Steve Ferritin, and I'm a nephrologist out of Houston. And I am very honored to be here today, and I'm going to be moderating this session. And I'm both a doctor and a patient. And so being that I have seen healthcare from the other side, I'm very, very interested in advancements in science. Because if the life cycle or if the natural history of a disease can be intersected by advancements in technology during our lifetime, it's a win-win situation for us. And AAKP has always been at the forefront of innovation. And a lot of companies, you know, they have their niche. But one of the things about AAKP, starting back with dialysis itself, was always at the forefront of innovation. And so we're going to be sharing some of the exciting innovations that have been happening with you, and hope as they can continue that you, your friends, and people you know, and of course our patients can also benefit from them. We're very graciously happy to have the sponsorship, the very generous sponsorship of some of the companies that you've heard about today: Merck, Rockwell Medical, and Sandoz. And without the aid of our sponsors, it's very, very difficult to put presentations and conferences together. But at the same time, these companies are wonderful in that they actually solicit and want the benefit of what a patient has to offer. So they tailor what they do around the needs of the patient. And this is critical for our profession and for the health industry and for the renal community at large. So I just want to give a special thank you to the generous sponsors. The presentations that we're going to talk about, I'm going to start with talking about the life cycle of a drug. It's going to be followed by Rob Tierney, who's going to be talking about a new way to treat your anemia. And then Jim McKay is going to be talking about an overview of biosimilars. And that's going to be followed by me, and I'm going to be, I'm not with Merck, but I'm going to be stepping in on behalf of Merck. But I'm not going to be talking specifically about the Merck product. I'm going to be talking about hepatitis C and sort of where we're going with that. And I think you'll find that will be helpful. Rob is the founder and CEO of Rockwell Medical. He has been a sales manager for Dial Medical in Florida. They were acquired by Gambro. And Rockwell is a fully integrated biopharmaceutical company. They target both the dialysis. I'm very proud to be using their products in the dialysis unit, and then they also target the CKD market. So they basically cover the range of kidney disease, the spectrum of kidney disease, and they have products for iron deficiency as well as secondary hyperparathyroidism. And of course, they have a product that we use in our dialysis facilities called Citrapure. Jim McKay has been with Sandoz now for 3 years, but he's no stranger to working with clinical research and medical affairs. He's been doing that for at least 8 years. He is now the Director of Clinical Development and Medical Affairs, and he's part of the clinical development team that's working on biosimilars in the United States. He works with stakeholders, payers, physicians, and patients and legislatures when it comes to understanding the role that biosimilars plays. You're probably asking, what is a biosimilar? Keep asking that question, and that way you will know a lot about it after Jim has spoken. And I'm Steve Ferritin. I've been involved with AAKP for many, many years. I chair the Medical Advisory Board. I am the editor, along with Chris Blagg, of Renal Life. In my other life, I teach at Baylor, and I'm a clinical professor at the university there. Not a real professor. I'm actually on the side, but I still get the wonderful opportunity to interact with med students and fellows and residents. And I'm also a medical director of our dialysis unit and in private practice. So I've been around the dialysis world since the 1970s, and I'm very happy that I went the route I did with nephrology. We're going to be talking about a very interesting thing right now that's the life cycle of a drug. And the objectives of this talk or that at the end of the day, you're going to understand a little bit more about what the FDA is and what a research cycle is. You're going to understand how a drug progresses through its life cycle from inception to patent to research, and then what happens at the end of its exclusivity period. And you're going to have a basic to formulate how pharmaceutical industries work and why drugs cost as much as they do. Now, you're not just going to understand this from me alone. You're going to understand it, I think, from being here. And I think it's very important for you and for all of us to understand this better. So we're going to be talking about the different phases of drugs. Now, the question that we all ask is, well, what does hypertension have to do with this talk? Well, everything, in the sense that in 1898, people didn't know a lot about high blood pressure. But somebody did discover back then that it was related to the kidneys. And then in 1939, they discovered that there was something going on, that the kidneys were making a product and it was going to the liver. And then they came up with the term angiotensinogen. And ultimately, 1954, ironically, the same person that invented a type of dialyzer, who also invented a machine that would automate how we do blood tests, the autoanalyzer, Leonard Skeggs, he figured out that there was an enzyme reaction that developed an active form of this angiotensin and made it angiotensin 2. Now, what happened next was just fascinating. One of the fellows went down to South America, and he was visiting his family in Brazil, and he went out in the Amazon, and he noticed there was a snake, and every time the snake, a pit viper, would bite an animal, a lizard or something, that animal would instantly die from low blood pressure. And so he figured out this must have something to do with what we're doing up in New York. So he got together with John Lara and with Vane and all of these guys. He said, I think we've got this product that is an angiotensin enzyme blocker. It's fascinating what's going on here. And they took it back, and Vane at this time was going to work with Squibb. So they developed this drug, and after they developed it, they noticed that you couldn't take it by mouth. It was the famous SQ14225 that was coming out. There was Ceralacin was another drug. And they noticed that you couldn't take it by mouth. So what they did was they figured out how to come up with an enzyme in the stomach that would block the breakdown product. And when they were finished, they ended up with SQ14225. There was 881 or something before that. So this all it had was a number. And then they figured out that this drug was going to be very valuable. They filed the patent on it in 1976. And in 1977, the name Captopril was born, and it was approved. Now listen to this, it took several years, but it takes even longer now. It was approved by 1981. So the FDA approved the patent for Capitan. And in the FDA, which has the power to do all this, wasn't born because people just said, okay, we need an FDA, you know. Every time the FDA came out with something, it was because there was usually something bad was happening. And 1912, they were noticing that they were giving morphine-laced soothing syrup for infants, and the infants didn't do that well on the morphine, and so there were some laws pertaining to that. In 1930, they gave it its name, but in 1938, there was a company called Massengill, and they took this product called sulfanilamide, and they said, well, we'll make it easy to swallow for children. So what we'll do is we'll combine it with this product that's out there they're using for antifreeze, ethylene glycol. The problem was that it went down really well, but 106 kids died. So that's when the Food, Drug, and Cosmetic Act was passed. In 1962, there was a drug that a sleeping pill that sort of woke everybody up called thalidomide. And after a lot of birth defects, the Kefauver-Harris Amendment was born, and that really got the FDA working on their concept of safety and effectiveness. There have been several other acts that have been pro and con. There's a lot of controversy. But the bottom line is the FDA has a very important role, and it's a very important that we respect the FDA, but at the same time, there's a lot of pressure they're under that contributes to healthcare costs. So there's several ways that a drug comes to market. And the first phase is after people understand that, there's this snake venom out there, what are we going to do with it? They start doing research on animals. But after a while, they say, okay, it's time to look at this in humans. So they go with healthy volunteers. And what they do is they characterize the pharmacology of the drug, and they figure out what the drug does and what it's going to do, and then they help to design future studies. And this is the phase 1 period of a drug. These are called phase 1 studies. The next thing that happens, and of course this is healthy volunteers, and they look at kinetics and mass balance, areas under the curve, and this is sort of an example of where they go with this. And there's a lot of things that happen during this period. They want to know what the drug does to the body and what the body does to the drug. And this is where a lot of answers come out on whether you eat it with meals, how much to give, how long it lasts, and how it's metabolized. So this is a very important phase. But the next question is, well, okay, and you can see how much this contributes to the drug label. The next question is, well, what is it really going to be helpful? Are we just coming up with this drug? Is there value to it? So that's where they come up with trying to figure out whether there's value to the drug or not. So they start looking at this with several hundred patients. These are multi-center studies. They can be open label or closed label, and they may last a short time to maybe a couple of years. And they want to know if the patients have a statistically significant response. This is important for the pharmaceutical industry because it's during this period that they determine whether their drug is going to even go to a phase 3. You'd be surprised how much money is spent on drugs, how many millions of dollars, and then the company says, we're just not going anywhere with this drug, let's scrap it. And unfortunately, the companies have to eat those costs. So, but ultimately they say, okay, is this drug effective? Is this drug really, really going to help people? That's when they come to phase 3. Phase 3 can be thousands of patients. These studies are blinded, which means the investigators don't really know what they're doing with the drugs. Now, a lot of the investigators like me, we don't know what we're doing anyway, but blinding us so we have no idea what we're doing with these drugs is really a good thing. So we're not biased, and we can just have an objective way of using these drugs. The question is this drug going to be beneficial? And that's what everybody wants to know. So the long-term studies are done. Adverse effects are documented. And once the drug passes stage 3, the company goes to the FDA, and I said, I think we have a product here, and we need a label, and we need your approval. And so they go to the FDA. They have to obviously work with the FDA to get a label made, and then once it's approved, the companies are going to stick with that label. And there's a lot that happens, with respect to drugs being on label and off label. That's a totally different lecture. And the bottom line, though, is that the drug, once it gets FDA approval, they have a patent on the drug, and they're allowed a certain period of exclusivity. And I believe it's 7 years for an orphan drug. A new chemical will have 5 years. Other drugs have 3 years. So exclusivity, and Jim's going to correct me on some of this stuff because he's going to be a lot more aware of this than I am. But the bottom line is the exclusivity period is very, very important for the drug. Unfortunately, during that exclusivity period, they may have trouble with their manufacturing. There may be things that hurt. That can hurt the way that the drug is able to recoup some of its cost. So this is a summary slide that will show you sort of how this business goes from when they start their original application to when they actually get their, what we call the NDA, their new drug application that is submitted. And they have an investigational application, and then they have the NDA. So you can see what happens. Then the drug is going to be used, and then the phase 4 can be either marketing or fine-tuning, or they can do studies to look for other new drug applications, and we're going to talk about that too. Now, this is what the drug companies want to happen, that you spend a certain amount of money on your drug, but you're able to recoup your drug price later on. And it's critical that they do this. Unfortunately, if that early number is too deep and too much of a volume in the early drug and they don't ever recoup their drug, then the company is going to lose money. When a pharmaceutical company loses money, that's not good. And so we want to make sure that, you know, and there's a lot of problems with this, but we're not going to solve them all today. But the bottom line is drugs are very expensive because a lot of times they have to recoup some of these costs. Now, the good news is sometimes a drug will come out and they will show that the drug is valuable, but they'll also find it's good for other things. For example, captopril came out and it was great for lowering the blood pressure. But people like Ed Lewis and stuff with the collaborative study group also showed that this drug would be valuable for treating diabetes. It was also shown to be useful for strokes. And same with some of the other drugs. A lot of times people come out with other drugs that are similar to the drug that comes out with originally. For example, captopril was short-acting, so somebody came up with a drug that lasted 24 hours, 25 hours. So now that's the drug that's being used right now. A company in Europe came out with a drug called perindopril and was very aggressive in doing research in Europe on this drug as well, and found out the value of their drug for strokes. Along with a diuretic, I will have to add. Ramipril also came out and showed their value of their drug with protein. So you have a lot of drugs that are me too, and ultimately, as most of these drugs get on the market, the price of the drug goes down. In phase 2, we might uncover new side effects, but we might uncover in phase 4 that the drug may also have other benefits. And you can see that poor thalidomide, it was started out as a sleeping pill. They found out it was useful for leprosy, and the guy doing the leprosy research found out that if he gave it to some of his leprosy patients, they would get a good night's sleep. But they also figured out that they could use the drug later on for myeloma. And so as it turned out, you know, thalidomide now has an FDA indication for myeloma. Aspirin, great drug for fever, baby aspirin in babies. They found it would really be a poison in babies ultimately for liver disease, Rye syndrome, but it was really great for preventing strokes. So now some of us take an aspirin every day for a stroke. So you can see how some of these old drugs are recycled and they have new uses. And some of these uses, as long as they're FDA approved, the companies can promote those. There's a lot of controversy about the pharmaceutical industry. There's a lot of books that have been written about the good and the bad and the ugly. The bottom line is, I'm just going to highlight, the good is that the drugs have increased our life expectancy. We're now living in the 70s and 80s. That's good. I can say that, and getting into that era of my life. The bad is it takes forever to develop a drug, and there's a lot of new ideas out there, a lot of innovation. I'm part of an oncology philanthropy. We're doing a lot of things with markers and stuff where we're going to shorten that time period and also shorten the cost. And then the ugly. Drugs are just too expensive, and there's a lot of finger pointing. There are a lot of people that are just upset. They don't maybe think straight, and they come up with all kinds of nasty things. So that's the good, bad, and the ugly. The bottom line is blame is always a problem, and this isn't a blame problem. This is a systems problem. So I just want to end on a happy note that one of the drugs, the drug that we were talking about in the beginning, ended up being useful for diabetes, and now it's sort of the standard of care to use an ACE inhibitor to manage patients with diabetic nephropathy, that, and also its cousin, the angiotensin receptor blockers. So I'm getting out on this note, and thank you very much. I believe you're next. Okay, Jim is now up next. Thank you.
Don't worry. I got it. All right. All right, well, thank you. That was a very informative presentation. I'd like to thank, my name's Rob Chiowini. I'm the CEO of Rockwell Medical. I'd like to thank the patients and AAKP for having us participate in your conference. Rockwell Medical is an innovative leader in developing and commercializing products for dialysis patients. I started Rockwell 21 years ago, and when we started with concentrates. These are, or dialysate, the product that runs through the artificial kidney. It's a solution, cleans your blood during your dialysis treatment. And over that time period, we launched 8 new products, and 4 of those became the standard of care. We're a very innovative company. We're always trying to find ways to do things that will give patients a better way of life. We've got some exciting products, and we believe that these products will help improve your lives. And so I encourage you later on today or tomorrow to visit our exhibit booth and get some information on those. Our most exciting product is called Triferic. And Triferic was recently approved by the FDA, and it's available now. And it fits this topic very well, this life cycle of emerging therapies. It's a new revolutionary anemia treatment. And if your anemia is treated, you're going to feel better. You're going to have more energy. And this is our top priority right now, and it's to help patients get access to this drug. Because when we think about the life cycle and we get to the market, there's still another step, and it's to get the proper reimbursement. And we'll talk a little bit more about that. So when we think about the life cycle, we've got to identify a need. Someone's got to come up with an unmet need or see an unmet need or a product that's out there and make an improvement. And then we've got to look at it and say, okay, is this viable? Does it work? And like the doctor mentioned, you're generally going into phase 1, phase 2 study at that time. And we discovered this technology in 2002, late 2002. We started clinical development in 2004. And after 10 or 11 years of clinical study and $150 million and going through the process, we were fortunate to get FDA approval because it's usually not such an easy thing to do. And so we've done a lot of heavy lifting. We've got a lot of work done. And now we've got to get patients to have access to this drug. And we want to educate patients as well. That's one thing we've also put a priority on. And so what is Triferic? Well, everybody here has probably seen this picture. And we all know when we're on dialysis, we're getting stuck with some needles. We've got some blood trapped in the lines, the dialyzer, we get some lab draws, and so we're losing blood. Every treatment, we're losing a little bit of blood. And it's 156 times a year. Now what we know is how much iron is in that blood. It's 5 to 7 milligrams on average for every patient, no matter who you are, where you dialyze. You're going to lose about 5 to 7 milligrams of iron, and that adds up. It's about a gram a year. And there's nothing that's been available until now to replace that. And Triferic's innovative because it's delivered through dialysate. And so now when the patient sits in the chair for the 3.5, 4 hours, and they lose that 5 to 7 milligrams of iron, Triferic puts it right back in. So you're made whole during the treatment. And just like we have calcium and magnesium and potassium cross the dialyzer membrane and enter your blood, we now have iron, just a little bit and just enough to replace what you lose. And that's innovative. What makes the drug really special is what it does when it enters your blood. And this slide has a lot going on, but what we have here is a picture of transferrin. And transferrin is the taxi cab in the body that carries iron to the bone marrow, where you need to get iron to help generate red blood cells. And what's special about this drug is as soon as it crosses the dialyzer membrane and enters the blood, within seconds, it binds or donates its iron completely to transferrin. And it goes right to the bone marrow. Doesn't get stored in the liver, doesn't go anywhere else, it goes right to the bone marrow. And it's very similar to how we get our iron from food and maintain our hemoglobin. Now, when we contrast this to what's been available for the last many years to try to treat anemia on the iron side of the equation, we see we've got IV iron. And IV iron is a repletion therapy. It was approved by the FDA as a repletion therapy, not a maintenance therapy. But it's been the only thing out there to use. And it's very different. All IV iron products are encased by a carbohydrate shell or have a carbohydrate moiety. And as a result, when they enter the blood, instead of binding completely and immediately to transferrin, they metabolize in the liver. And that's all right. Some drugs metabolize or designed to metabolize in the liver. The issue in dialysis, and it's similar in cancer, is we have an inflamed patient. There's a lot of inflammation with chronic illness. And so what happens is the iron gets to the IV iron gets to the liver, and then it gets trapped in the liver because of inflammation. There's a peptide called hepcidin that mobilizes and blocks the iron from coming out of the liver. So as we've been giving IV iron to help treat anemia all these years, the majority of it has been getting trapped and stored in the liver. Just 2 to 5% actually will bind to transferrin when it's injected. And as a result, we've had an increase in our ferritin levels. And the blue bars here represent ferritin. And you can see, since the 90s, how the ferritin level has gone higher and higher. Now, what is ferritin? Ferritin is a marker in the body of our stored iron and inflammation. And so today, the average ferritin in the US is 4 times higher than it was in the 1990s when IV iron was approved. In the 90s, when it was approved, the average ferritin in a hemodialysis patient was right around 200. And the indication for IV iron was to treat iron deficiency anemia, which is defined as a ferritin of less than 200. And it all made sense. If the ferritin fell below 200, you give an injection of IV iron, you bump it up over 200, and then if it comes back down, you give another one. But as you can see over time, for many reasons, including the mode of action of the product, the ferritins now are, on average, in the US at 800. There's no other country in the world with ferritins this high. And if you look at these red dots that follow the blue bars in lockstep, you'll see that those represent hospitalizations from infections. And there's a lot of published data that shows the association of IV iron and infections. And the infection rate's risen 43% over that time period. So what does this lead us to? Well, we have what's called in our population, our dialysis population, functional iron deficiency. And that's when we've got iron in the body, but we can't mobilize it. We can't get it to the bone marrow. And that occurs, from what I said a minute ago, it's because we have an inflamed population. And when there's inflammation in the body, hepcidin mobilizes and blocks most of that iron from coming out of the liver. The average is 5 to 7 milligrams lost per treatment, as I mentioned in the beginning of the slide deck here. That's about 1 gram a year. But if you look at DOPS data, you'll see that the average amount of IV iron given to patients is 3.6 grams, almost 4 times higher. When we look at the two products, we can see the differences. We've got one, Triferic, that's specifically designed to deliver just a small amount of iron every treatment, and it's an iron that binds the transferrin immediately, gets to the bone marrow, and gets used up. There's no iron trapped in the liver. The ferritins don't increase. They actually come down over time when patients are on Triferic. There's no inflammation, no infections. Importantly, there's no anaphylaxis. Now, there's anaphylaxis with IV iron. It's not rampant. It's a percentage, a small percentage, but it happens on occasion. And that's because of the carbohydrate moiety. And to put this in perspective, a typical dose of IV iron, it usually occurs weekly, sometimes less, is about 100 milligrams. And if we were to give the same amount of iron as that one single dose that happens when you're on treatment, it would take you 20 treatments over 7 weeks. And that's the difference between the two products. So when we look at the safety, the important points here are there's a bunch, but there's a couple that really stand out. One, this was the largest safety database ever submitted for a parental iron product. A lot of patients, a lot of patient years of exposures, a lot of doses. And what we saw was that the adverse events were similar to placebo. And that's very good. The other thing that was that stood out, was very important, was we saw a decrease in blood transfusions of greater than 50% versus placebo. Now that's important because if you're on dialysis, the way you get out of dialysis is you get a kidney transplant. But if you get a blood transfusion, a kidney transplant, the odds of it being successful or getting one get reduced considerably. So it had excellent safety. Now, I want to show you quickly. I'm going to see if I can do this right. I think I got it. A video.
My name is Charlene. I am a teacher, a singer, an actress, a choreographer, and of course a mother. The first time I had dialysis was in the hospital. I was going to the doctor and she said, get her to the hospital now. It was, you have to have a pacemaker, you have to have a catheter. We're talking about a fistula. They just said, oh, we're going to dialysis now. It was instantaneous. It was the first time in my life where there were no choices. That was frightening for me. My life has always been busy. I've never been a still person. I just want to do so much. There's so much out there to be done. And I get depressed when I don't have the energy to do stuff. And when I went on dialysis, I was always tired. Always tired. I'm gonna go home and I'm gonna go to sleep. What kind of life is that? You know, that's no kind of life. And when you don't have the energy, you become very depressed. Because I'm on dialysis, I can't do what I used to do before. And then when you get Triferic, it's like opening the window. Suddenly, I can do these things. I'm not falling asleep all the time. I suddenly got an infusion of energy. We have a choice of how we can feel while we're on dialysis. With Triferic, you feel like you're getting a little of your life back. I can tell the difference in my body with Triferic and without it. Without it isn't a place I want to be ever again. Triferic helped me. It did more than help me. It gave me a better lease on life.
Okay. let's see if I can get back to my slide deck. Oh, here we go. I got it now. All right. So I've got one more slide. That was Charlene. And when we went through clinical development, we went in front of an AdCom, an advisory committee, because iron has never been given this way before. I mean, it's truly revolutionary. It's something that's never been done. The way we've given iron has been the same way for 25 years, IV. But we now have a product that can do it better. And she was in the clinical study, and we had a lot of patients in the clinical study, and we would get letters from investigators that patients would give them because some of them were concerned. The study was blinded, but they were convinced that they were on the drug because of things like this. And they were concerned that they were going to be pulled off the drug while we went into the FDA approval process. And so she contacted us when she found out we had an advisory committee and said, look, I want to go. I want to talk publicly and make sure that this drug gets approved. Now, I can't sit here and tell you that this drug is going to affect everybody on dialysis the way it did Charlene, but what I can say is that it makes a lot of sense that if you're losing a little bit of blood every treatment over time and it doesn't get replaced with iron that can go right to the bone marrow, chances are you're going to feel tired. And if you get a drug like this, chances are it will make an impact in your life. So when we think about the life cycle, and we see a drug like Ferric, and there's other drugs that are going through these life cycles, that are getting approved, and now they're going to the commercial market, maybe the most important last step is, and especially in dialysis, more so than many other areas of medicine, is reimbursement. And when we think about the life cycle and a partnership with patients, one of the things we've done at Rockwell is we've made a commitment to try to get the word out to patients, to work with patients, to find out what are some of the problems and the issues. And we hear them, but when we come to a conference like this, we get a lot more information. And is there something we can do? Is there something we can create? You may have an idea. It might be something as simple as a Band-Aid or a fistula clamp or something. And then if we identify it and we say, okay, this makes sense, we make the commitment to spend a lot of money and a lot of time to get approval. And then once we do that, we come back around and we're still in partnership because we've got to secure the proper reimbursement. Because if the proper reimbursement isn't secured, unfortunately, you may never see the drug and be able to experience the benefits of it. And so in our world, CMS plays a big role in shaping innovation and access to products. If the proper reimbursement isn't secured, your access won't happen. And companies like Rockwell and Sandoz and some of the other ones, Merck, they won't make the commitment to spend hundreds of millions of dollars and bring a new technology to the market if they know there's no money for it when it gets there. And so AAKP is a great conduit for patients. As Paul mentioned in the very beginning, they can take the message to the right people and get the right things done, but it takes part of the patients as well. So my point is it's a partnership. And if you want more information, we've got a nice website, we've got an exhibit booth, and I think I'm about out of time. Thank you.
Jim.
Seamless. Okay. Thanks, Dr. Ferritin. Thanks to the AAKP for the honor of being here and talking to you today about biosimilars. So the title of my talk is an overview of biosimilars. How many in this room have heard of biosimilars before? Biosimilars. All right, so very few people. One there, a few. Okay, good. So that's good. So it's good that I'm here explaining them. If you have questions during my talk, I think we have a question and answer session at the end, but you can interrupt if you'd like to. Okay. So drugs can be divided into roughly 2 large categories. Okay, we have on the one hand what we call small molecule or synthetic drugs. These, as the name implies, can be made, man-made in a test tube or in a laboratory. And of course, for production for treating patients, we do it in large manufacturing plants, but they're completely synthetic. They tend to be very small drugs, and they're completely reproducible. So every time we make a batch of this type of drug, we can make it exactly the same from batch to batch. Another type of medication is the other type of medication we call biologics, and these are much, much larger molecules. So large, in fact, that it's impossible for us to make them synthetically, like I described for these small molecule drugs. So the way we make these, we actually get nature to make them for us. We get cells to manufacture these drugs, and then we purify them and we can use them in patients. So two very different approaches to manufacturing, 2 very different types of drugs that we use to treat patients. With biologics, because they're made by living organisms, they're not exactly the same every time they're made from batch to batch. There's slight modifications or slight amounts of variation from batch to batch. For a synthetic drug, Dr. Ferritin was talking about the life cycle of a drug. We have a research and development portion. That's followed by a review by the FDA, and hopefully at the end of that point, the drug is approved for use in patients. The drug then gets a certain amount of exclusivity, as Dr. Ferritin said, where it's the only drug on the market. Once the patent expires, then other manufacturers can make this same drug. This is referred to as generic medications. With generic medications, there are multiple versions on the market. The competition drives down price. That's the main benefit to the patient and to other payers of healthcare, is that the price goes down. That's a fairly easy process with these small molecule synthetic drugs. Not only is it possible to make exact versions each time a batch is made by the same manufacturer, when a different manufacturer makes a medication, it's also possible to make the exact same version. So a generic is exactly the same as the brand medication. With biologics, however, because of the way they're manufactured, being manufactured in cells, and because of the inherent variability of the molecules, it's not possible to make an exact copy. But what manufacturers and developers of biosimilars are able to do is to match the variability of the brand product or the reference product. And that's why instead of being called generics, and identical copies of the reference product, they're called biosimilars. So that's where this name biosimilar comes from. Okay, as I said, for these large complex drugs, we actually use cells. We use nature to manufacture the drug. The way that works is we have what we call a host cell. We insert into that host cell a gene that tells the cell how to make the protein or how to make the drug. We then grow the cells in large bioreactors and then purify the drug of interest through a relatively complex purification process. The purified biologic is then put into its final form, which can be a syringe or a vial. Usually has to be stored under refrigerated conditions, and the shelf life is relatively short for this type of medication because, again, it's made from living organisms, sort of like something you keep in your refrigerator like milk. It doesn't last forever, so the shelf life tends to be a little bit shorter. So a more complicated drug, more complicated manufacturing process and purification process, and then the finished product is also more complicated in the storage and delivery. More precautions have to be taken with respect to storage and delivery. Okay, so we have these 2 different types of drugs, and there are 2 different statutes that give the FDA the authority to approve these 2 different types of drugs. Dr. Ferritin mentioned the Food, Drug, and Cosmetic Act, which was passed in the 1960s. The predecessor of that actually dated back to the early 1900s. This is a pathway that governs the approval of these small molecule drugs. In 1984, it was amended by what was called the Hatch-Waxman Act, and this created a streamlined route through the FDA for the approval of generic medications. So since 1984, we've had a relatively easy process for getting generic medications approved. About 80 to 85% of prescriptions written today are filled with generic medications, and generic drugs save the healthcare industry and patients and payers about $140 billion a year. So they've made a very substantial impact on the cost of healthcare in the U.S. The other statute that's used by the FDA to approve this other class of drugs, biologics, is called the Public Health Services Act. And it also has a history that dates back to the early 1900s. So all biologics that are approved in the U.S. are approved through this Public Health Services Act by the FDA. The first of the modern biotech drugs, which fall into this classification, were approved for use back in the 1980s, and those patents are just now starting to expire. So in 2010, the Public Health Services Act was amended to create an abbreviated approval pathway, which now allows the FDA to approve biosimilars. So these are follow-on versions of biologic drugs that are approved once the biologic drug goes off patent. And it allows for a less expensive version of the biologic to come to the market. So this will increase competition on the market and will drive down prices of these very important and sometimes very expensive drugs. So the law is very specific that it informs industry the requirements that they have to meet in order to get one of these biosimilars approved. The FDA definition is shown here. It's a biologic product, so made from living cells, that is highly similar to a reference product. So you notice it's not identical because of this variability that exists in the reference product. So the biosimilar has to be highly similar or match the variability of the reference product. And it also cannot have any differences except for minor differences that are not clinically meaningful or differences in clinically inactive components. Okay, so it's a very strict law, it's very stringent, and there can be no clinically meaningful differences between the original biologic and the biosimilar with respect to safety, purity, and potency. So biologics are a very important class of drugs. Interestingly, they represent about 8% of prescriptions now, but they account for a much larger percentage of overall spend on prescription medications. This is a list of the top 10 revenue-producing drugs in 2013 and then what's predicted in 2018. And as what's indicated by the blue highlights is that 7 of the top 10 revenue-producing drugs are these biologics, so made in living cells using biotechnology to make them. Some of these may look familiar to you. For instance, Humira is advertised on TV quite frequently now. It's used to treat serious diseases like rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis. These are very serious diseases that, left untreated, result in significant mortality and have a very large negative impact on the lives of patients that have these diseases. Lantus, of course, used to treat diabetes. Enbrel, you may have seen Phil Mickelson on TV. He takes Enbrel for psoriatic arthritis, another disease similar to rheumatoid arthritis. Some of these biologics also treat cancer. For instance, Avastin and Rituxan are important biologic drugs that treat different types of cancers. Again, these are expensive drugs. They can cost $50,000 to $70,000 a year for patients that are on them. The first of these drugs were approved in the early 80s and again are starting to go off patent. And so this amendment to the Public Health Services Act was just passed in 2010. And as these expensive drugs go off patent, the FDA now has the authority to approve follow-on versions, which again are these biosimilars. So at Sandoz, we have a fairly large pipeline of biosimilars in development. We actually have the first biosimilar that was approved in the US. It was approved in March of last year, and it's a biosimilar to a biologic called Neupogen. Neupogen is a drug that's used to stimulate white blood cell production. It's used by patients undergoing chemotherapy, and a side effect of chemotherapy often is suppression of white blood cells, which leaves the patient susceptible to infections. So they would take a white blood cell stimulator to protect them against infections and to allow them to continue their chemotherapy. So the first biosimilar on the market again, approved last year. It was a biosimilar to this white blood cell stimulating factor. 2 other biosimilars have been approved recently. One is a biosimilar to Remicade. It's manufactured by a company called Celltrion and will be marketed in the U.S. By Hospira, which has recently been bought by Pfizer. And Sandoz, we recently had our second biosimilar approved in the U.S., and it's a biosimilar to Enbrel. The Remicade biosimilar and the Enbrel biosimilar are not on the market yet. We have a 6-month period where we have to notify the original manufacturer before we can start manufacturing or start marketing our biosimilars. In addition, there are some legal issues that still have to be resolved. So exactly when these drugs come on the market remains to be determined. Humira is set to go off patent next year, and there are several companies that have biosimilars in development for Humira, as well as all the other biologics on this list. So again, just to recap, we have these 2 classifications of drugs: small molecule, chemically synthesized drugs. The follow-on versions that come to the market when those drugs lose patent protection are called generics. The other main category of drugs are these biologics. We use living cells to produce them because they're very large and complex. And because of living cells that produce them, there's an inherent variability, so we can't make exact copies. So we don't call them generics, we call them biosimilars. They're analogous to generics, but different, and they're treated different legally. Biologics are complex drugs that treat very serious diseases like rheumatoid arthritis and some types of cancer. Also anemia. Neupogen is a biologic that treats anemia. The act that the FDA uses to approve biologics, the Public Health Services Act, was amended in 2010, and now the FDA has the authority to approve follow-on versions of biologics, which are called biosimilars. And we have 1 biosimilar on the market now. It's a biosimilar to Neupogen, which stimulates white blood cell production, and many more biosimilars in the pipeline. And the promise of these biosimilars is that they will increase competition and reduce prices of these very important and very expensive drugs. That will improve patient access to these important drugs, and will also free up money in the healthcare system to pay for new therapies that are coming in the pipeline of many pharmaceutical companies. Okay. Thank you.
Okay. How do I get out of this thing? Okay, now my eyesight's not that good. Let's see. Where's my hepatitis C?
Scroll over this PowerPoint right there, it might pop up.
Right there?
Yeah.
There it is. That's why I like your Travatin. I like your glaucoma medicine. Okay, let's go into play. I think that's F5. How do I get that Rockwell thing off? Oh, it doesn't matter. Okay. Today I'm going to continue the cycle of talking about the life cycle of drugs by speaking of a drug, a group of drugs that's used to treat hepatitis C. And you saw the biosimilar, one of the biosimilars there. This is a very important medication, and I think those of us who have been in practice for a while recognize how devastating hepatitis C can be. One disclosure, I'm a consultant to a company called Athlon, and also Merck is a sponsor of this meeting. This slideshow, however, was developed by me, and I'm solely responsible for its content, so this is not developed by Merck or any other pharmaceutical company. And I need to say that by law. The objectives of this study are that at the end of this talk, I think we should all be more familiar with the signs, symptoms, and laboratory analysis and the clinical course of hepatitis C. Know the differences between the genotypes of hepatitis C. But more importantly, we will have learned that therapy is genotype specific, and we will have heard the different new therapies for each of the genotypes and also each of the disease pictures that the drugs are useful for. And so we'll start at the very beginning. First of all, people want to know what a virus is. And when they think of a virus, I'm sure they think of their computers now. But it's actually a piece of genetic machinery that's surrounded by a little shield that'll protect it. And it derives its structure and ability to replicate from the host cells. And in some instances, that might be your liver or somebody's liver or somebody's inside us. And it basically takes all of our nutrients and it uses those to package its wall and its RNA, and it continues to replicate and divide itself. And in the end, it goes viral. And so that's where we get that term. It's actually not invented in Silicon Valley. Viral RNA makes proteins that make up its shell, and it replicates itself very effectively. So this is sort of a schematic of, or a picture or a cartoon of what a virus looks like. It's got its envelope on the outside. It's got its core, and the core protects the DNA, the RNA, and it's not DNA, but it's actually an RNA in this molecule. And the core is sometimes called a capsid. And the cell has a life cycle. In other words, it invades, the virus will invade our cells, and the RNA will actually incorporate itself into an area of the cell called the endoplasmic reticulum, for those who want to know its name. But what it does, it incorporates itself into that part of the cell and starts making proteins. It just sort of takes over. And it takes over the cell. It makes the proteins, but the proteins that it makes are its capsid and its envelope and its own RNA. So it basically makes itself, and then it takes over the cell, and then it goes out to another cell until it spreads all over the body. And so this is all it is. It's just a simple protein, group of proteins, and it's controlled by a little strand of an RNA. Some of the RNA controls the structure, and some of it controls the duties that has to happen. So it's a very simple process. And what, Steve? Panel needs to take questions. What? Okay. Wait a minute. We have 5 minutes left. Okay. All right. Well, basically, we're going to talk very fast. Okay, we're going to go into high gear. The bottom line is that this type of drug is capable, several drugs are able to fight this disease. Well, basically, there are several types of hepatitis that we're not going to have time to go into here. The main type that we're going to be talking about is hepatitis C, which comes from blood transfusions. Hepatitis C can be measured by a variety of tests. There are 130 to 150 million people around the world with hepatitis C. So needless to say, since about 700,000 people die, new medications are important. And you can see that hepatitis C is something that is back on the rise. Many patients can have no symptoms at all, but some can recover and not be symptomatic till the very end. Unfortunately, many patients can be carriers of the disease. So it behooves us to try to get rid of this disease. It's initially asymptomatic, but like I say, it can go on to lead to cirrhosis. You can get it from needles and toothbrushes and razors, and you can also get it in dialysis. So we in the dialysis community are very interested in drugs that can stop cirrhosis. I can't talk about cirrhosis right now just because of time constraints. The genotypes, there are several different types the hepatitis C. They are numbered from 1 to 7. The goals of the therapy are to give people about a 12-week course of it and see if in that 12-week period we can have a lasting response. They talk about a response lasting over 24 weeks, but we really want a cure. And that's because the disease can cause liver cirrhosis, it can cause cancer, and it can also interfere with kidney disease and cause a glomerulonephritis. There's been a long course in the evolution of therapy, but the bottom line is we are now at a stage of drugs that there's a combination group of drugs. There's about 5 or 6 of them out there. Zapatier is one that works with subtypes 1 and 4. And the advantage of some of these drugs is their effect on some can affect the kidneys, and some can work on the kidneys, and some can work in patients with kidney disease. So some of the drugs can be useful in patients that are actually on dialysis. And the reason we're talking about the drug Zapatier is that it actually can be given to dialysis patients. These drugs are very, very expensive. Luckily, Zapatier priced theirs to be at a much better price point as other drugs. But needless to say, these drugs are expensive. Some of the drugs are more risky than others. The Zapatier molecule has a less risky safety profile. Some are more effective, like Sofosbuvir, which you saw mentioned in a previous talk. It can be more effective in treatment-naive patients. Some can be used in cirrhosis patients, and likewise, some can be used in dialysis patients. I can't go over, because we don't have that much time, the side effects, but we can certainly talk about this at another time or off-label. The summary is that hepatitis C is a chronic disease that affects 130 to 150 million patients, and that we now have medications that can actually treat this disease and cause a lasting cure almost of this disease. Prescriptions are based on the disease process and the genotype and whether the patients have kidney disease. Again, I want to thank our sponsors, Merck, Rockwell Medical, and Sandoz. And the last session is going to begin in a very few minutes. We now have a few minutes, seconds maybe left for questions and answers. I apologize that we went over. Yes? Hepatitis C is diagnosed through blood tests. You do a screening test, and then you do a more specific test for that.
Can you have what is it, when you have a shot to take and eliminate the chances of you ever getting it?
There is no vaccine for hepatitis C. There is for hepatitis B and even A, but there's none for C. Somebody needs to ask questions to the other speakers as well.
Is there any possibility of those prices for the Hep C meds being made more reasonable?
Oh, gosh. I wish. You're asking the wrong person. Unfortunately, in other countries, they are cheaper. This is a very lengthy discussion that might involve a lot of people beside me. Richard. Somebody else.
I just want to be clear on it. Are you saying that using that during the dialysis process makes it such that you'll feel better from not losing as much iron?
Oh, come on up here.
No, what I'm saying is, if you get your iron replaced, that should lead to an improvement in treating your anemia. So if you're going to, ultimately what will make you feel better, I mean, there's a lot of things that will make you feel better, and the doctor's better to address all those things than me, but I know one of them is less inflammation, and I know another one is if you're getting enough iron, as well as you'd have to get enough ESA to create healthy red blood cells and treat your anemia.
Is this something that would change, or would be included in the bundle? I'm just trying to get my head around.
So Right, so the drug is in the bundle. And what we're doing is we've been working with CMS to get the proper reimbursement, which is called transitional add-on payment. And when CMS put the bundle together in 2011, they took all the drugs that were being used at the time, they took their average sale price, and they allocated a certain amount, $5, $50, $60 per treatment. They built a one-payment bundled reimbursement that came out to about $245. What they didn't do, and they acknowledged it at the time, was they didn't account for any drugs that weren't yet on the market. And so there's still not a pathway for these drugs, and until there is, they need to look at each individual drug and be able to give it that transitional add-on payment, which lasts for about 2 years and helps the providers adopt a therapy, and that helps them get the therapy to the patient so the patients have access to it.
Okay.
Yes.
Yes. It's a bloodborne infection, so it can travel from blood droplets, it can live a short time on gowns and equipment. There's a lot of ways, and be sure to visit the CDC exhibit because they will go over a lot of reasons why bloodborne infections can be transmitted in a dialysis unit and why safety is so critical there. Okay.
I was wondering, I had heard in a little bit that the reason the drug prices are so high here and less high in some other countries is because, like I think the European Union and even the Canadian government has laws that puts floors on it, and it ends up that Americans end up paying for the research. I mean, I've heard that analysis. Do you think that's true, or is that so?
Well, the short answer is yes. Any other questions? I guess not. Okay.
Well, thank you for to our speakers, and we're going to take a short 5-minute break so we can get the next group on.